All right, let's take the words that accurately fill in this concept map. Here in Lesson 4, we learned that the human genome can be divided between the non-coding DNA and the protein-coding DNA. The non-coding DNA represents about 98% of our genome, whereas protein-coding DNA represents about 2%. No matter how you divide it, you've got 2 copies of the human genome in every cell. Now, non-coding DNA includes 3 main types of non-coding sequence that we discussed. One type was a promoter, another type was an enhancer. Now, enhancers are bound by activators, so that's how we knew enhancers went here instead of here. We know that if promoters and enhancers are 2 of the types, then the third type are silencers. Silencers are bound by repressors, and activators and repressors are types of transcription factors. Transcription factors specifically bind around the TATA sequence, which is essential to drive initiation of transcription via the promoter. So everything in this lesson really centers around the promoter sequence, because the promoter is the main effective non-coding DNA region that regulates transcription of a protein-coding gene. Now, transcription factors generally are proteins that bind any regulatory region of DNA. Activators and repressors are very specific types that bind certain regions, like enhancers that promote transcription initiation via promoters, and repressors that bind silencers, which silence or inhibit transcription from a promoter region. But no matter what, promoters sit in front of protein-coding genes, which are essentially the divisions of protein-coding DNA here. Now, our transcription factors that bind regulatory regions of DNA thus regulate the expression of protein-coding DNA, or genes. We also learned very specifically that just like in Lesson 3, that different sequences of protein-coding DNA can be called alleles, the same is true for non-coding DNA. We can have different sequences called alleles, and these all originate from mutations, which we'll learn about more in Lesson 6. Overall, in the human genome, both protein-coding DNA, even though it represents only about 2% or less, and non-coding DNA, which represents the vast majority of our genome, they work together to specify thousands of different cell types. And together, these are organized by the process of development that makes us who we are, all of our complexity, all of our traits.